So we are ready for our second speaker from this morning's session, who is going to be joining us from Denmark, from the Technical University of Denmark, and this is Erwin Skoof. He first became interested in single-cell protein analysis when he was a postdoctoral fellow, and he started adopting our methods, both the use of isobaric carrier from SCOPE-MS, and more recently, he's also adopted the use of freeze heat cycle for lysis of the cells that we introduced with the minimum sample preparation and SCOPE-2. So I'm really delighted to see that Erwin is using a methodology that we've developed very successfully and applying it to interesting biological problems. And in particular, he is going to talk today, I believe, about the hematopoietic system in the context of malignancy. I look forward to hearing about your latest results, Erwin.
Thank you, Nikolai. I hope everybody can see my slides.
Yes.
Perfect. And indeed, thanks for the kind introduction, and indeed, all praise for making this meeting happen among the current circumstances. Really a shame I couldn't be in Boston this year, but hopefully things will have improved even more next year to have the meeting fully in person. But indeed, I'm very thankful to be able to present some of our work here. Indeed, very much inspired by the work that you have done over the years, and we're kind of trying to implement it and optimize things. And as you talked about yesterday, there's so much room for improvement and boosting the performance, and we're really happy to be part of this community of the single-cell proteomics. So today I really want to talk about using bulk single-cell proteomics for characterizing heterogeneity, indeed in the hematopoietic system. And I will mainly be talking kind of on three main topics: a bit of background, then how we've used single-cell proteomics towards detection of heterogeneity, and then finish on some current developments that we've just pre-printed earlier this week. So why are we actually interested in single-cell? I mean, I don't think I need to spend too much time on this, but for those who are less familiar with the blood system, we know that it's really a hierarchy with our long-term and short-term hematopoietic stem cells at the top. These are what maintain the blood system throughout your life. And then we have the myeloid and the lymphoid branches with lots of unique subpopulations within them, all serving different functions. I mean, classical assays with flow cytometry and gene readouts really started defining these populations, and xenograft assays in mice. But it was more the recent years of single-cell technologies, and in this case, single-cell RNA-seq, where we really saw, okay, this HSC pool is actually much more diverse, much more heterogeneous than we initially assumed. Also going down the hierarchy is much more diverse, and in fact, it's actually much more of a developmental continuum rather than really discrete cell states. So we absolutely need single-cell technologies to define these cell populations and to understand these differentiation processes. So we're not only interested in the normal blood system, but also very much in leukemia, which is also, apologies, hierarchical in nature. Here we have the leukemia stem cells at the apex, and these can self-renew, i.e. they can make more leukemia stem cells, and they can differentiate into progenitors and blasts. And it's actually these blasts that kill the patient. It's also these blasts and progenitors that chemotherapy targets. The leukemia stem cells are quiescent and therefore not susceptible to standard chemotherapy. So if we want to start eradicating leukemia in vivo, we need to gain a much better understanding of the cell population. And that's why we're so interested in single-cell proteomics. So indeed, we take basically the same approach as SCOPE-MS, using a carrier or booster channel in the 126. And then with 16 or now 18 plex, we label single cells with individual TMT tags. So I won't talk much on the process of that. At the moment, we're using a lot of the Explorers 480 technology with the FAMES iMobility setup. We've built a computational pipeline that was briefly touched on Monday, and I'll go in a bit more depth on what it does and what it's useful for. And we really envisioned this kind of integrated experimental and computational workflow that allows us to visualize our data, show our single cells, how many proteins we saw, and kind of the specific protein expression levels within each of these cells. So really, indeed, based on a lot of previous work that was published, but trying to implement it in our own way. So talking about how we prep these samples, I mean, we use a fax depositing setup for throughput reasons mainly, where we can select populations of interest and deposit single cells into a 384-well plate. We add a bit of lysis buffer, a microliter of 20% TFE, to kind of help the lysis process and denaturation of the proteins. Then we boil the plate and we freeze them. And then using our Dispendix I.1, which is a non-contact liquid dispenser that can go down to 8 nanoliter. So there's still room for improvement in terms of downscaling the prep. And we use this for the trypsin dispensing and the TMT labeling. So especially in TMT, it's very important, also shown by Nikolai's group, that you have to randomize your pools of cells, because otherwise your data interpretation will be flawed subsequently. And doing this with a automated liquid handler really helps a lot in doing it automated, not making mistakes, and keeping track of your setups of your cells. I think that's another feature that we were really keen on implementing by using the fax machine, that we actually record the cell surface markers that we stained the cells with for subsequent data integration. So after digestion and TMT labeling, we use the OpenTrons OT2 to basically pool the samples, add our booster channel or carrier channel, and then we can speed vac the samples and run them without additional cleanup. And this was recently published. So to test our workflow and to establish everything from scratch, we wanted to see if we could detect heterogeneity. And we thought this AML hierarchy, the acute myeloid leukemia hierarchy, was a good model system for this. And thanks to my time in the John Dick lab in Toronto, we had access to this sample called OCI AML 8227. It's a patient identifier, so it was a relapse sample from the patient who basically got treated but relapsed and came back to the clinic with more severe disease. And if you look at the CD34, CD38 profile of this, we have the bulk of the population, 90% of the cells, which are 34 negative. This is what we call the blasts. And then the 34 positive, 38 negative in red is what we deem the leukemia stem cells. The proof for this being that if we isolate these cells, we plate those cells in culture and we wait 7 days, we can see that the whole hierarchy gets recapitulated, including more LSCs, we get progenitors, and we get the whole blast population. Whereas if we take the 34 and 38 positive, the progenitors, and plate those, we only see the blast cells appear and maybe some progenitors remaining, but very crucially, no leukemia stem cells. So this, in our opinion, really proves that this is the leukemia stem cell fraction. And more importantly, it's a heterogeneous cell population that we can culture in vitro without serum. We just add a lot of growth factors, and therefore represents a nice system for validating our single cell proteomics workflow. So to test it first, we basically did pre-enrichment of cells. So we sorted blasts, progenitors, and LSCs, and we plated, or we sorted 24 injections worth of cells, i.e. 2 384-well plates in total, to inject at 2 separate injection times. Because we know from Chris Rose's work, published a while ago, that you need long injection times. We also know it from the scope work, of course, but we weren't sure how much injection time was really needed, especially on the FAMES generation of instruments. So we figured, let's first test our kind of mass spec parameters to establish a workflow that could give us good data and lots of proteins to play with, hopefully. So just to give an overview of what happens when you inject at 300 milliseconds or 500 milliseconds, you can see the signal to noise in the single cell channels is definitely increased with the high injection time. This comes at the cost, of course, of number of proteins we identified per cell in individual injections. So this is without any imputation. And that to reach this 1,000 proteins per cell, we need to inject only at 300 milliseconds, because otherwise the scan time gets too high. And of course, we do have the missing value problem that all single cell technologies deal with, which is actually better at the lower injection time because you simply get more peptides. But of course, protein numbers are only one thing. We also wanted to see how well do we separate our cells using more advanced methods. So for example, if we take UMAP as built into our receptor pipeline, we can see how well do we separate out the blasts from the progenitors and LSCs. And in fact, how well do we separate these progenitors and LSCs, which we know functionally are very similar, but molecularly should still be different. And we can see here the clear advantage of using the high injection times of 500 milliseconds, as we are getting a much nicer separation of the LSC and progenitor populations here. So we can overlay the number of proteins we identify for each individual cell, and you can see that we kind of visually can represent these numbers from the table. So in terms of quantitative accuracy, because it's of course very important to measure, we wanted to quantify the ability to tell apart progenitors and LSCs and measure that with a silhouette coefficient. And here you can see again, the 500 millisecond is definitely better at separating progenitors and LSCs. Whereas if we compare to bulk data, i.e. standard proteomics of the same cell culture system, where we sorted out the 3 populations, and with GMT Pro just compared them at bulk level, you can see that if we use all our proteins, we get quite decent correlation with bulk data, definitely better with the 500 millisecond than the 300 millisecond. And if we take the top 400 proteins, we can see that we are very well matching the bulk proteomics level of quantitative accuracy. So to help us interpret all this multidimensional data, we built this pipeline called Scepter, Single Cell Proteomics Readout of Expression. So Benjamin, a PhD student I'm jointly supervising, he really spearheaded this. So it's a collection of Python scripts. You can download it for yourself and play with it, and it's tightly integrated with ScanPy, which I guess is safe to say the leading algorithm in the single cell field for data interpretation. We can normalize our data without having to define which cell populations we're dealing with. So instead of having these batches of TMT reporters, once we normalize it in Scepter, we actually get our much nicer cell maps representing the different phenotypes. We can filter faulty wells, so to say. So either maybe 2 cells were sorted, no cells were sorted, something went wrong with the reagent dispensing. Because we can see these spikes up here occurring in some of the wells that clearly have too much signal to noise. And therefore, when we filter these out based on the some signal to noise, we get a much more kind of consistent cell signal coming out. And we can visualize that also on these plots, kind of separating out the number of proteins per cell and the sum signal to noise. And we can set these boundaries of which cells we deem to be trustworthy. And if you wonder if we can find the empty wells, yes, we can. So if we incorporated in our experiment empty wells that just get the trypsin and the TMT tags, but no cells, we can clearly see them separated out from our real single cell wells. And therefore, these we argue that these cutoffs are actually a good way of making sure that you're using reliable data. And then finally, we can, of course, also impute the missing values between cells. So just to give you a bit more visual representation of what you can do with Scepter once you have your data, I mean, as I mentioned, you can overlay your FACS data. So if we base our UMAPs on a protein level quantitation and we have our separation of cells, and then overlay the FACS recorded data from the index sorting, we can see that indeed the progenitor LSCs are 34 positive, as we know from the culture here, and that the blasts are 34 negative, and also that we have this enrichment of 38 positivity only in the progenitors and not in the LSCs, as we expect. So we can ask the software what proteins are different between our populations of interest, so we can find perhaps cell-specific or cell-enriched proteins. We can then overlay those protein expressions on the single cells in our original UMAP embedding and do some pathway analysis for functional interpretation. So then we were keen on figuring out, can we actually use this to detect heterogeneity? Because with single cell RNA-seq, we know that with 10x and those kind of high throughput platforms, we just throw a lot of cells at the problem without any pre-selection and just try to let the data tell us what was there. And therefore we argued if single cell mass spec can actually detect cell heterogeneity, we should be able to distinguish bulk sorted A2 to 7 and recapitulate that leukemic hierarchy that we know exists. So to do this, we sorted 6 384 well plates, a bit over 2,000 cells in total, and identified just under 3,000 proteins across all these cells. So of course, blast being the dominant population in the culture, 2,000 cells were blast. We got, almost 80 LSCs and a couple handfuls of progenitors. And then our first question was really, is this sufficient to start seeing kind of some differentiation trajectories? And the state of the art for this at the moment seems to be pseudotime analysis, where you find your very primitive cells here, which is basically a differentiation trajectory with your most differentiated cells at the other end of this U shape. And of course, again, we can overlay our FACS markers and confirm that our primitive population is 34 positive and to some extent 38 negative. And then of course, we have a 38 positive progenitors as well. And then at the end of the trajectory, we have our 34 negative blasts. So this seems to suggest that indeed the pseudotime orders the cells according to the differentiation trajectory we know from our experiments on mice on what the cells do functionally. Of course, I wanted to take this a bit further. So if we then annotate our populations onto this, we can clearly see our LSC and progenitors being located down here, but actually we can start seeing the separated trajectory of now 38 positive blasts in red and 38 negative blasts in blue, potentially suggesting that there's actually two differentiation paths contained within this pseudotime measurement. So if we then visualize the protein heat map of this trajectory, so we order all the cells according to the pseudotime, we can start finding these clear clusters. So we have kind of the primitive cluster of proteins here, and then differentiating out to the more differentiated blasts. And kind of want to point or draw your attention to this cluster 3, because here we can actually really separate the 38 positive and negative blasts. So this third cluster here seems to be a key trajectory specific protein cluster, separating out functionally these two blasts. And that suggested to us that perhaps there are two differentiation pathways kind of from indeed LSC through progenitor through 38 positive blast to 38 negative, or perhaps there's one directly going from the LSCs to the 38 negative blasts. And to validate this, we wanted to test this functionally. So we basically, thanks to the help of Colleen, sorted out the LSCs and the progenitors and plated them separately. And then at day 2, day 4, and day 10, did a global CD34 and 38 profile. And we can see that already on day 2, the LSCs have formed these 38 negative blasts, started perhaps forming some progenitors already, and a few 38 positive blasts, but that the whole hierarchy actually only really starts appearing at day 4. And that to us really suggested that yes, there are actually these two differentiation trajectories in LSC, and this would have never been picked up with bulk proteomics. So can we stress test this setup even more? Because of course, a model system is nice, but we want to apply this to real biological questions. And kind of something that we've always been wanting to do is basically healthy human bone marrow to really try and decipher cellular trajectories within this hematopoietic hierarchy. So I just blinded out the populations we didn't focus on, but these 6 populations were those that we actually sorted out in an enriched form, but also in a bulk form. So we sorted in total 721, just 34 positive bulk cells, which contain all these populations, and then 192 pre-enriched cells. So this was really a proof of concept experiment to see whether this is worth pursuing. So we ended up with 32 pre-enriched cells from each of these populations. And if we see this embedding here, we can see a very nice spread and perhaps some cellular trajectories already appearing here. Number of proteins suggested that technically the experiment works also on really true primary cells, which tend to be smaller, tend to have less proteins expressed. So we're very happy to see that our workflow does or is able to analyze these types of primitive cells. And then really, if we use the pseudotime again to try and rank these populations, it was very nice to see that indeed HSCs are at the apex, MPP are also high, but start becoming into this branching point. And as we go down to MLP, we go further down this branching point and CMP. But then really GMP and MEP are very separated. And that's exactly what we see here. The MEPs are a separate branch, the GMP are a separate branch. And it was nice to know that even with just 32 cells, we were already starting to see these kind of cell trajectories. And this is why we're now doing this in a much larger scale. So Nil and Benjamin are working on this quite hard. We can again use these protein heat maps to figure out what are more cell enriched populations. So do we have our kind of HSC signature here? And can we actually compare, for example, the HSC to GMP versus the HSC to MEP path? I mean, I don't have time to talk about all these proteins, but we can see some differences among these proteins in terms of how they separate these populations, which seems to suggest that we can find at least some of the proteins that are involved in the cellular differentiation trajectory. So I mean, for this part, I really hope that I was able to show you that we have an experimental and computational workflow at hand that is able to detect cell heterogeneity and separate cell types, that it works both on pre-sorted, so pre-enriched cells and bulk sorted cells in a discovery mode. It's semi-high throughput. We try to automate as many steps as possible, both for accuracy, but also for making sure that our people don't get bored pipetting too much, which is a challenge with these types of large-scale projects. Scepter is a good way to quality control and normalize your data, and eventually visualize and extract the biological meaning, which is the whole point of doing these experiments. We need to filter out outliers, and that can be done with Scepter. In our experiments, and I didn't have time to go in depth, but you can read it in the paper, we didn't seem to need a reference channel or an additional empty channel because we can spot these with our filtering. And the normalization based on reference didn't seem to add any quantitative accuracy. And in the paper, we also show that we can integrate both balanced and unbalanced data sets. If we don't know which cell types are present and therefore probably end up with an unbalanced setup, we can use Scanorama, which is a single RNA-seq tool to integrate these data sets and make meaning of them. And indeed, we confirmed that long injection times are desirable for accurate quant, and that 500 milliseconds seems to strike a good balance between proteome coverage and accuracy. So now with 18-plex, we can increase our throughput to 16 single cells. And we're trying to push our chromatography to kind of get more like 20 runs done per day rather than only 8 to 10, and therefore, potentially be able to reach 320 single cells per day. So then we were fortunate enough late last year to take delivery of 2 eclipses. We called one Everest, and the other Himmelbjerg. So all our instruments in the facility have mountain names. So Himmelbjerg is the highest mountain in Denmark, whereas Everest is the highest mountain in the world. And depending who you ask, they say one is more magnificent than the other. I guess you have a good view from both of them. But indeed, we're very pleased that we got access to this technology to really see if we can be more sophisticated in how we run these instruments. So I want to spend the last 10 minutes or so talking about MS2 level analysis, MS3 level analysis, and a new method we introduced called reticle MS2. So I mean, very briefly, we posted this on bioRxiv earlier this week. So if you want to have a read, please do, and feel free to let me know what you think. So standard, we run MS2 analysis where you do an MS1 scan, we just do a top N sequence, and then you read out that the MS2 in a very long injection time, high resolution Orbitrap scan, where you identify roughly a third of your scans. So you're wasting a lot of time. Then the tribrid setup enabled this MS3 analysis, which gets rid of co-isolation. So you do a quick scan in the linear ion trap, so low resolution but high sensitivity. And then you select precursors from that MS2 scan to read out at high resolution in your Orbitrap. This works well, but you are again spending a lot of time on scans that you didn't identify at the MS2 level. So with the Eclipse, Thermo introduced what's called real-time search, meaning it searches these MS2 linear ion trap scans and only selects those that it was able to identify into a peptide and selects those for MS3 in the Orbitrap. So you're spending only these long injection times on those that you've already identified. So we thought, well, can we actually leverage this technology and do a similar setup that we do a quick MS2 scan in the linear ion trap? And we were amazed to see that we only needed 23 milliseconds of injection time. So it really speeds up your scan speed and we can find more features that way. And then instead of doing an MS3 scan where the precursor selection causes some signal loss, do an MS2 scan, but this time in the Orbitrap at high resolution. So we again use real-time search to select those that need to go for the high resolution scan. So I mean, why did we call it reticle? Well, it stands for Real-Time Search Enhanced Quant of Single Cell Spectra. It's a great website if you're looking for acronyms. When we saw this, I wasn't immediately aware what a reticle was. But it's actually a scope or a microscope piece. So it's a really nice way to kind of home in the instrument on those peptides that you want to look at in high resolution. So we thought it was quite appropriate. So of course we wanted to validate this and to start this and to kind of set up the method, we figured it was wise to use a diluted sample rather than real single cell. So again, we took this OCI AML 827 sample and had triplicates of blast, LSCs, and progenitors spread across a lot of the channels. And then again, with this booster/carrier channel in the 126. This was all done on the 50 centimeter micropack chromatography. So just to give you a background idea of the performance, I mean, we compared MS2 at 500 and 750 milliseconds. We compared real-time search MS3 at 500 and 750. And then the reticle MS2 at same injection times. So what's immediately apparent is that we have a lot more quant spectra in the unbiased MS2 500 millisecond. And of course, we decrease due to the higher cycle time in the 750. Whereas in the real-time search ones, we get very similar amounts of quant spectra between MS3 and reticle. However, what's interesting is that the identified quant spectra, i.e. those quant spectra that actually gave us a peptide ID, it was really reticle 500 millisecond that stuck out as being the best performing, even outperforming the shorter injection time MS2. And the difference between reticle and real-time search MS3 is the fact that the MS3 level analysis uses the ion trap spectra for peptide identification, whereas reticle, you use the Orbitrap spectrum, both for quantification and identification. And indeed, you get this massive boost in identification rate compared to standard MS2 when you use the real-time search as expected, and therefore leading to single cell, quote unquote, because it's diluted down to a single cell level. We breached this 1,000 proteins per cell with 500 millisecond and basically approached with 750. You might wonder how come that this 500 millisecond method works so much better with reticle than without. And we argue that partially the reason for this is that we can do this closeout, meaning that we can tell the mass spec, once you've identified 4 peptides for a particular protein, stop analyzing it. We can set the limit to any limit. So we tested 4, 10, and just turning it off completely. And you can see that the 10 peptides and no closeout are very similar in terms of how many proteins we identified with a particular number of peptides, and that indeed those 2 methods identified a lot of high abundant proteins, most likely with basically very, very many peptides that won't improve your quantitation value. So how well do these methods separate our populations? Because again, we should be able to separate In this case, the blast versus the LSC. So we again use the silhouette coefficient. And here it was suggested, so if we use only those proteins identified in all the approaches, it's very clear that the MS3 with the very high injection time has the best performance. And then the reticle with the very high performs second best. And then we decrease significantly for MS2 in terms of separating these cell populations. Again, this difference between reticle and MS2, you can see in this closeout setup that we simply get more high peptide per protein, kind of in this median range from 6 to 10 or 11 in the reticle method, whereas the MS2 really has a lot of these 15 plus peptide proteins. And this is even more strong in the 500 millisecond data set. So this is really a nice feature that we can use real-time search for. Then of course, we just wanted to do a cross comparison between reticle 750 and all the other methods. So each box here is comparing reticle 750 to all the other methods. And we can see that it outperforms MS2 easily, the 750 reticle method. Performs similarly to real-time search MS3 500 milliseconds. So you can see that benefit of the higher injection time for reticle MS2 than we were able with MS3 here, but that MS3 indeed seems to be the most accurate method here. But then what about accuracy versus precision? I mean, we need to assess these workflows in both methods. I mean, you can be very precise. You're very good at measuring the same thing with the same values, but you're actually quite off the real value. So we want this high precision and we want the high accuracy. And the accuracy you can measure by comparing to bulk proteomics. And here indeed, we can see the big benefit of MS3 by getting rid of the co-isolation. We're measuring more accurately, whereas the precision, it's basically if you inject the same sample 3 times, what is your coefficient of variation? And here again, MS3 works very well, but we also see that the reticle method is performing virtually identically, if not even slightly better due to a lower spread of the box plot. From these data, we concluded that MS3 is more accurate, but the high injection time of 750 millisecond on reticle without losing proteome depth results in better precision. And this became much clearer when we started analyzing real single cell samples. So we also subjected more than 100 cells to each of these methods, and then could see that our average protein count per cell, only in the reticle methods, was really up around this 1,000 proteins per cell mark. And then, of course, it just shows that real single cell is a different beast than doing dilution series. So it's not bad performing in terms of numbers, but of course we would like to get more proteins, more markers out. So in terms of UMAP separation of these populations, you can see that while we are able to separate these 3 cell types, so the LSCs or the progenitors, the LSCs and just the blasts, but that clearly the reticle 750 millisecond has the nicest kind of condensed cell populations to showcase its separation power, and actually even better than the real-time search MS3. And this just visualized differently in the silhouette coefficient that again, reticle methods are even outperforming MS3. And we kind of hypothesize that it's simply the protein coverage that gives us more proteins to do the clustering on. As you can see here, almost 2,000 proteins in reticle 750 compared to 1,200 proteins in MS3. And that kind of suggests that yes, you also need the high proteome coverage to get good separation. So with that, I mean, I hope I showed that reticle provides an improved MS2 level method for deeper per cell proteome coverage. And we actually also get better signal to noise due to this 750 millisecond injection without losing proteome depth. So while the accuracy is not as high as MS3 in terms of absolute quantitative accuracy, but the deeper proteome coverage and joint high injection time is more precise and therefore should allow us to detect better biological differences. So the real-time search is really essential. I mean, without real-time search, we're at about 500 proteins per cell in a dilution experiment. So really only half the proteins, I would say. And now we're focusing on trying different chromatography options, such as the EvoSep Whisper platform and the MicroPack limited sample column for additional benefits. We're really wanting to push the throughput in terms of decreasing the gradient length and also lowering the boosting ratios to be able to not have as much noise added to the system. So I mean, just as a last teaser, I mean, if we're using the 20 samples per day Whisper 100 method, so this would allow 320 cells per day. With reticle, we are actually approaching this 1,000 proteins per cell, whereas with standard MS2, we're just at above 850. With the 10 samples per day, the 2-hour gradient, we are already above the 1,000 proteins per cell with MS2. And with reticle MS2, we actually started approaching the 1,500. So that was quite exciting. And then thanks to pharmafluidics and Selenium, we've been testing a lot the limited sample column, and we had a nice demo. Thanks also to Angeli and Fabiana and Claudia and Carl Mechtler to kind of help us demo this system in our lab. And you can see that without any booster, with MS2, we were getting about 400 proteins. With reticle, we were getting 700 proteins. In terms of peptide coverage, we were improved with reticle. But especially here at the signal to noise in the single cell channels, you can really see this massive increase in quality of the quantitation, jointly due to reticle, but also very much due to the proteome chip. So with that, I really want to thank a lot of people, I mean, especially Bo, my former postdoc mentor, who really supported me in kind of getting this going. Benjamin, of course, and Neil, the 2 PhD students closely involved. John for the former postdoc mentorship in Toronto, Ulrich at DTU, who's been really supportive. Lots of people at Thermo, who we've been collaborating with intensely over the last 2 years on real-time search and the Eclipse. EvoSep for their help with the Whisper methods, pharmafluidics with the limited sample, of course, the DTU proteomics core, our other technology partners, and funding. And thank you, I'd be happy to take questions.
Thank you, Erwin. We have a few questions in the chat, and unfortunately only 2 minutes left from the time slot, but I think we can push it a little bit and also make sure that we have some time for the break. And then perhaps you can answer some of the questions in the chat as well. So I'm going to ask 2 questions from the virtual audience and one from the in-person one. So one is, do you have matching single-cell RNA sequencing data for the BLAST 38 positive and negative cells? And how does that compare to the single-cell protein data?
Great question. I mean, we just got some data through collaboration with John's lab. We haven't looked in depth in kind of really head-to-head comparisons. We haven't looked at the BLAST population yet. What we saw in the LSC was that we could see these quiescent populations both at protein level. We found kind of quiescent stem cells and cycling quiescence or cycling LSCs that were going to differentiate into progenitors and blasts. And we also found the similar 3 populations in the RNA-seq data. So indeed, we want to validate, do we see this whole differentiation trajectory that we think we saw at least also at the RNA level, or if that's really a protein specific, which would of course be a nice argument why you need protein level data.
Is reticle open to earlier versions of the TriBERT?
Unfortunately, as far as I'm aware, real-time search is only for Eclipse and upcoming generations. There are some special agreements that might be available for the older generations that you can make with Thermo, but as far as I'm aware, it's Eclipse and upwards only at the moment.
Yeah, Erwin, great talk. I really enjoyed. I would like to go back to the situation where you had the 2 cells population, which was very hard to do separate, and then you did separation by very low accumulation time versus the just standard MS2 where you have more scans. I think it would be very useful to see, can you separate those 2 population for those extra scans that you did not get for the low accumulation time? Just those eigenvector should either play a critical role world to be able to separate those two very close cells line, or just they basically been useless to be uploaded MS2 more data because they are not going to separate those. I think this is very critical moment to understand, is the extra data with lower quality actually helping with the very close cell lines or not? That's where the question, I think, wasn't answered clearly, neither in bioRxiv nor here. Just please look at that. I think it's useful.
Yeah, no, thanks. That's a great point. I mean, if I understand correctly, you're wondering if these ion trap MS2 scans would help, that never got identified, would help separate the cell types. Is that correct?
Yeah, basically, if you just take everything which the same and remove it and leave only those that you get on the straight MS2 scans, type of methods, and try to separate just those two close cells just simply on those eigenvectors, do not looking at anything else which was very well identified, and see if those are helping or not, because this is very critical sample. Do you need more data with less quality, or you just okay to set up with 500 proteins, which is I'm pretty sure RNA-seq people will not agree you with, but that's where the critical question is.
Yeah, thanks. We can look at that.
Hi, that was a beautiful talk. Quick question related to that is, for UMAP projections of your single cell proteomic data, do you calculate a subset of variable genes for conducting the principal component analysis before calculating the embedding, or is it on all the proteins that are detected? And how would that change across your two different injection times that you were comparing?
Great question. I mean, I'm not as familiar with all the details of the clustering as Benjamin is, so feel free to reach out if you need more detail. But indeed, we try to, for the comparisons between the methods, we make sure that we use only those proteins identified in all, so that we are comparing apples with apples, rather than just having a method identifying more and therefore separating better. So we impute the data the same way when we do that. It's the nearest neighbors imputation. So in the end, the data should be comparable. But yeah, I'm not entirely sure if I answered your question correctly.
So I'm really torn between my love for questions and my intention to stay on time. So I think I'll suggest a compromise. We'll still take part of the remaining break to enjoy the food and the coffee and the drinks outside. And in the meantime, our virtual audience can stay online, and you, Erwin, maybe can read the questions from the chat that you can answer and keep answering them during the break if you're willing to do that.
Absolutely. No,